Chapter 12 discusses DNA replication. It focuses on the basic mechanism by which DNA replicates, by which DNA doubles before cell divide. So we will start with discussion of this basic mechanism of replication, and afterwards we will examine different modes of replication. We will also talk about the requirements, what is needed for replication, which proteins and enzymes actually participate in the process of DNA replication. So what are the requirements for the DNA replication? Firstly, DNA replication has to be extremely accurate. Check 1. Check 2. It has to proceed with a high speed. Now, why it is? Why we need such an extreme accuracy when we copy the DNA? Firstly, you know that we all start as a single cell, as a fertilized egg which forms zygote. Zygote contains approximately 6.4 billion base pair of DNA. Now we know this one single cell undergoes millions of replication and forms the embryo, forms the fetus, forms the human being. So if only one error and million base pair occurred during this embryogenesis, we will end up with over 6,000 mistakes, mutation in every single cell division. So multiplied by millions of division, cellular division taking place during embryogenesis, and you can immediately imagine what a catastrophic outcome it will have. Secondly, even in adult age, our cells do not live forever. So they age, they die, and they have to be replaced by new cells. Or sometimes we damage cells, we cut ourselves. Those cells have to be replaced. If you cut yourself, you want that those cells will be replaced by identical cells. The replication of the cells, meaning replication of DNA, has to be accurate. If it's not, what might happen, you know very well, cancer might happen. So we really need accurate, extremely accurate replication. Why high speed? Well, replication has to proceed with high speed because we have millions of base pair, billions of base pairs. So if we replicate DNA, we don't want to wait for days, months that this DNA is replicated. We need sometimes to replace those cells very fast. Now, remember when we talk about E. coli in chapter 9, E. coli can produce progeny, new progeny in 20 minutes. If you take into account that E. coli has approximately 4.6 million base pair of DNA, it has to proceed fast. And it does indeed proceed fast. It replicates at a rate of 1,000 nucleotides per second. So, what kind of replication would fulfill these requirements? And the answer is semi-conservative replication, which is based on the complementarity of the nucleotides. During semi-conservative replication, which is outlined like here, both strands of DNA serve as a template which gave rise to the new strand based on the complementarity. So after one round of replication, you end up with two DNA molecules which both are hybrids. They have one old and one new strands. However, this semi-conservative replication which is accepted nowadays as a sole mechanism of the DNA replication was not actually accepted immediately. Initially, there were three models which were proposed: conservative replication, dispersive, and semi-conservative. So what is the difference between them? In conservative mode of replication, although both strands serve as a template, these two strands stayed aligned together. A newly synthesized strand, which are outlined in red, forms the second molecule of DNA. In dispersive replication, dispersive replication mode suggested that both strands break down, and basically fragments will serve as a template, and this fragment will afterwards reassemble, so each molecule would contain pieces of old and pieces of the new DNA molecule. Now, semi-conservative we already discussed. In semi-conservative way, you have original DNA molecule which afterwards opens. The hydrogen bonds are broken, and basically you have two strand original which serve as a template for the newly synthesized strands, which newly synthesized strand I label in red. So this would happen during new synthesis. So after one round of replication, you will have two DNA molecule which would compose, or which will be formed, which will contain, better said, one old strand and one new strand. Now, do you remember we talked about the DNA molecule, that two strands of DNA are complementary to each other, but they are not identical. So how two non-identical strands can give rise to the two identical DNA molecule. So let's just outline some sequence. Let's say that this molecule of DNA has this in this sequences A, T, C, G, C. So the second strand is complementary, so it would be T, A, G, D. Let me just rewrite it again. T, A, G, C, and G. So you see this strand and this strand, these two strands are complementary but not identical. So how they can give rise to two identical molecules of DNA? So let's outline what happened right here. So you have old strand, which is A, T, C, G, and C, and here it is T, A, G, C, and G. So when DNA replicate, it replicate based on complementarities. These are polymerases which assure accuracy and speed would add complementary bases. So here it would be T, A, G, C, and G. And in this case, it will be A, T, C, G, and C. So now compare these two molecules of DNA. They are identical. Okay, so complementarity assures that two strands which serve as template, even though they are not identical, they will give rise the identical molecule of DNA based on the complementarity of the nucleotide base. So how it has been proven that the mode of replication of DNA is semi-conservative? So this picture outlines the basic experiments that were performed by Mendelssohn and Stahl, which confirms the semi-conservative mode of replication. So Mendelssohn and Stahl worked with E. coli, with bacteria, and they really have been intrigued by the DNA replication. So in order to define the mode, what they needed to do, they had to develop a method how to distinguish between old strand of DNA and newly synthesized strand of DNA. And they took advantage of fact that DNA contains nitrogenous bases, and they labeled bacteria with isotype, nitrogen isotype N15. So they cultivated E. coli in medium which contain N15, heavy nitrogen, for many generations, until all DNA in all bacteria have been labeled with heavy nitrogen. And they proved it. They took sample of this bacteria, they extracted DNA, and they performed equilibrium density gradient centrifugation. And in this gradient, they saw that all DNA for one band heavy nitrogen band containing DNA with heavy nitrogen. So all original DNA has been labeled by heavy nitrogen. So what they did next, so afterwards they took this bacteria, they washed them and transferred them into media which contain N14, light form of nitrogen, light isotype N14. And after transfer, they cultivated the bacteria in this media and took samples after first round of replication, second round of replication, and third round of replication. After each round of replication, they took sample, they extracted DNA, and they again ran equilibrium density centrifugation, gradient centrifugation. And what they observed after first round of replication, basically it's right here. After first round, they again had only one band. But this band was slightly higher than in previous case. So this one band immediately ruled out conservative mode of replication. Why? If you recall, in conservative mode of replication, the original strands of DNA also they served as a template for newly synthesized DNA. They reanneal and they form one DNA molecule, and the newly synthesized strand, which I will label in red, will anneal and form second molecule. So this new molecule will be labeled by N14 and the old one will be N15. Okay, let me just label it properly with proper color that it's not confusing. So this would be N15. So you see, after first round of replication, you will have one band which will be formed by DNA containing heavy nitrogen and one band which will contain DNA with light nitrogen. So since they obtain only one band, conservative mode of replication was ruled out. However, they still couldn't distinguish between semi-conservative and dispersive mode of replication. You recall, in dispersive mode of replication, you have pieces of old and new DNA in one DNA molecule. So the DNA molecule contains both heavy and light nitrogen. So we would also have only one band. So the answer was given actually after second and third round. After second round of replication, what they observed two bands. This immediately ruled out dispersive mode of replication. In dispersive mode, you would have only one band. Recall, pieces of old and new in every single DNA molecule, so it would form only one band which would be mix of heavy and light nitrogen. So since I had two bands, this confirmed that the mode of replication is semi-conservative. And why it is? Look right here. So the original DNA was labeled by heavy nitrogen. Now when this DNA unwind, so each of this strand which is labeled in blue correspond to N15, would serve as a template, but new strand, because the bacteria now are cultivated in N14, new strand, which is outlined in red, will incorporate light nitrogen. So both molecules will have old and new strands, so both will contain heavy and light nitrogen. In second round of replication, each of this DNA molecule will replicate. So each of these two strands would serve as a template. So template which contain N15 is in blue, but the second strand will be already containing light nitrogen, so it would have both. However, the red labeled strand of DNA which contain already light nitrogen, which will incorporate light nitrogen. So we will have two kind of DNAs, one which would only contain light nitrogen and other which would contain still heavy and light. So this will form two bands. So these experiments clearly demonstrated that the mode of DNA replication is semi-conservative. When DNA replicates, every individual unit of DNA replication is called replicon. right here. Each replicon contains origin of replication. One replicon, one origin of replication. You remember, in order that DNA can replicate, it has to have functional origin of replication. It's a place where the replication starts. Now, even though the DNA replicates, all DNA replicates by semi-conservative mode, this mode can happen, the semi-conservative replication can happen by several different ways. It can be either through theta replication, rolling circle replication, or linear eukaryotic replication. Theta replication can be found in bacteria such as E. coli, which have circular chromosome. Under theta replication, circular DNA chromosome forms one replicon with one origin of replication. It has bidirectional mode and it forms two replication forks. Rolling circle replication you can find in plasmid. You remember in bacteria you have plasmid independent DNA which replicates by rolling circle replication, and also in some viruses you can find this mode of replication. And of course linear eukaryotic replication is found in eukaryotic organism, and it's very similar to theta replications with difference that theta has only one replicon, one chromosome, one replicon, while in linear eukaryotic replication we have hundreds and thousands origin of replication, meaning replicons as well. So let's now very briefly outline all three modes of replication. So we will start with theta replication. So where the name come from? It comes from the fact that electron micrograph of replicating E. coli chromosome, you see right here, resemble a letter of Greek alphabet theta. That's the name. So how does it happen? At the origin of replication, you recall we have double-stranded circular DNA, and at the origin of replication, DNA unwind, and as it unwinds, it produces two single-stranded templates for DNA, for new DNA synthesis, and forms replication forks. One replication fork, second replication fork. What does it mean? That if you imagine that this is origin of replication, so replication goes both way. So it's bidirectional. So as it continues, it forms what we call replication bubble. And replication bubble is actually a place where two old strands are unwound and where the new DNA strands are synthesized, and this continue in both directions. So basically there are two replication forks, and this synthesis of new strand continues until eventually two circles are produced. The rolling circle replication differ from other two, from theta and linear replication, in two basic ways. Firstly, it is unidirectional. It only goes in one direction. Secondly, it does create, or it does trigger, a break in DNA strand. You see, let's pretend that this is a plasmid, bacterial plasmid. It is formed by double-stranded DNA, and here is the origin of replication. Within the origins, the enzyme cleave one strand, so only one strand is cleaved. And this break in one strand creates 5' end and 3' end. You recall 5' end has phosphate and 3' end has hydroxyl group. Now the 3' end hydroxyl group serves as a base where the new nucleotide are added by polymerases, and the strand which was not cleaved, so the intact strand serve as a template. So as the new nucleotides are added to 3' ends, this newly growing strand displaces the 5' end. And this displacement and synthesis of the new strand continues until it reaches the end. When it reaches the end, the 5' single-stranded DNA is cleaved and displaced. Now, the cycle may be repeated many times. And how is this 5' end actually unwound and how it is pulled? It is something like thread being pulled of a spool, so it's rolling similar way until it is completely displaced. Whenever the 3' end ends up synthesis, afterwards it is cleaved and displaced completely. This linear DNA, which is released here, can circularize, and it can actually serve as a template for synthesis of complementary strands. So basically it's a very efficient way because you can yield many offspring's DNA, so you can generate many daughters' DNA molecule by rolling circle replication. A linear eukaryotic DNA and eukaryotic human chromosomes encounter big problem during DNA replication, which lies in the size. A human chromosome consists of approximately 100 million base pairs of DNA. If you compare it to 4.6 million base pair in bacteria, you can see the problem. So, and you recall that DNA replication has to be not only faithful, exact, but also fast. So how evolution solved this problem? So the solution was to create many origins of replication. So linear eukaryotic DNA has many origins of replication, therefore many replicants. So the replication in each replicant starts from origin. It starts at the same time in euchromatin, let's say, and it proceeds both ways, so it is bidirectional. So each replicant has two replication forks, and the speed of replication at each forks is approximately, it varies between 500 to 5000 nucleotides per minute. So this replication proceeds both ways until two replication forks or two replication bubble connect and fuse together and produce two identical linear DNA molecules.